that's all I'll be saying in Italian today. Although I'm honored and privileged to be here today to talk with you about, I think, a pretty hefty topic. But given that I only speak English fluently and quickly, I do wish I spoke Italian. I think I can handle it, how fast you guys speak, if I just started earlier. I might have missed my calling. So I am delighted, like I said, to talk with you today. I am the chair of the Scientific and Medical Advisory Board of the United Mitochondrial Disease Foundation. I organized what I hope is a successful international effort called the Mitochondrial Disease Sequence Data Resource. And I'm also a member of the Scientific Advisory Board of a new genomic initiative called the Genesis Project, as well as consult, but unrelated to today's topic. So I'm going to try and address the topics I've been given in three major sections. Very quickly, I'll try to explain to you how clinical care for mitochondrial disease happens in America, and then talk about how clinical care transitions into really human research, and how we define those boundaries and how we get people to participate, both the specimen repositories as well as the registries and data resources. And then I'll try and end in our time together with how the patient tissues and samples are so very helpful. You've heard some beautiful examples this morning in the science talks already, but how those clinical samples really facilitate research into diagnosis and genetic etiology, as well as into mechanisms of disease and therapies, which is where we all want to get to. So this is published data from a few years ago. I like to call it the pre-next-gen data, how we did things before next-gen sequencing became the very first thing that we did, which is where we're at in America right now. So this is a clinic that I started at the Children's Hospital of Philadelphia in 2007 or so, and in 2008 to 11 we did just a retrospective chart review of about 150 patients that we had seen from infancy till much older age. About a third of the patients I see in the Children's Hospital are adults. And that's because there really is a huge absence of adult physicians in the United States comfortable with taking care of and evaluating mitochondrial disease. And I'm happy to talk about more if that's of interest. But these are the reasons in general people came to see us. So in children, developmental delay was among the biggest. Seizures, low muscle tone, chronic fatigue, family history of mito disease, laboratory abnormalities such as high lactic acids, muscle issues, autism, lactic acidosis, and many more. We actually have just completed a survey of patients now, asking what symptoms they have, and we asked up to 35 symptoms and of about, in two different surveys that we hope to publish soon, of about 350 to 400 patients. And it turns out every patient with mitochondrial disease had between 7 and 35 symptoms. So, none of these were mutually exclusive. Most people came in with more than one symptom. And I'll just note that I think in the Italian registry, if I understood the slides, not the words, that were spoken, GI dysmotility was only about 5 or 10% of patients. It's the number 5 top symptom that we have. When we actually ask patients what symptoms they're having, very, very few don't have some sort of gastrointestinal problem. And so before we could do next-gen sequencing and we had to go through the candidates one by one, really we only had to define molecular etiology in 14% and a biochemical diagnosis in another 4%. A large gray zone of it could be mitochondrial disease, but we weren't sure. And a fairly sizable number of people that came to see us that absolutely were not mitochondrial disease. As a matter of fact, in almost 10% of cases, we could prove what the other genetic diseases were, where it really had nothing to do with the mitochondria. This is just an example of how very many mitochondrial DNA mutations you can find if you start to look, and the ones in red are the ones that people traditionally, at least in America, would have considered to be on the common mutation panel. So when I started training in the early 2000s, if you didn't have one of these, you didn't have mitochondrial disease. Of course, now we know that's not true, but unfortunately, some people still think that that's the case. So whole mitochondrial genome sequencing is now possible and really expected. Plus, we are in the midst of many variants of potential pathogenicity, and there is no good way to confirm these clinically in the States, so that it relies upon research to make things such as cyber lying to somebody's goodwill. There is no clinical diagnostic test or order. And for example, we have so many patients with ATP 6 or ATP variants, and no way to test complex 5 function clinically in America, that we're now in the middle of a big project just to understand which of these variants could be truly pathogenic. This is just an example of many other diagnoses that were made besides mitochondrial disease, so many other metabolic and neurologic diseases and chromosomal abnormalities as well. So I think there was a slide earlier about using high-resolution SNP arrays to look for copy number alterations, and we absolutely agree, especially if there's developmental delay. So I think this group is on board that gene-by-gene diagnostic testing is very limited in its success. It's not that there can't be phenotype-genotype correlation, it's just that it's so excessive now, it's so beyond, I think, what should be the expectation of what a human brain can recognize, all the possible permutations, that it really is unsatisfying to test one gene at a time. And that's where the concept in America of the diagnostic odyssey, you test one gene at a time as it comes out for 10 or 20 years, and you still don't know the diagnosis in the patient. And so the value of the clinic is that, of course, a mitochondrial center or a clinic can recognize classic phenotypes, can guide optimal diagnostic testing, and doing it this way, of course, it takes a lot of time, and there's too many people. There's under 1,000 metabolic specialists in the entire United States. Many of those are retired or retiring, and so there's just not enough people to see these patients. So mitochondrial disease diagnostics has absolutely changed with next generation sequencing by doing either panels of testing, which a lot of laboratories are doing, although to be truthful, they're often just running exome sequencing and reading out the genes that you ordered. But the data is being generated because it's simpler for the laboratories. In one test now, if you do the either just the mitochondrial disease genes and the mitochondrial DNA genes, you diagnose about 50% of all of suspected mitochondrial diseases in one test. It's better than anything that's ever preceded it. Plus, if you then consign the data over, once it's been generated from the clinical setting to the research setting, it very much enables rapid gene discovery, of new genes. I always tell my patients, only about 5,000 or so of our 20,000 pairs of genes have been linked to any human disease. And so there's a lot of possibility that your gene, the one that's causing your disease, is in the other 15,000. And it's happening to us very frequently now that the day after we pull back the data and release the filters, we find it. And we often believe in family-based sequencing because from a computer bioinformatics perspective, it very much allows us to narrow in on the right gene. And this has really changed the way we view mitochondrial disease. So of course, we still view it as organ problems, multi-organ system dysfunction. And we know for the last 60 or 70 years that it's due to an energetic problem. But now we realize, as you heard again this morning, that it's all in which pathway is disrupted to get to the understanding of why you are sick and how we might help you. And so there have been different studies over the last few years about how efficacious doing different combinations of gene testing is. Avansi Muta's group showed that if you just sequence 100 known mitochondrial disease genes, you get about a 23% diagnostic rate. If you look at the mitoexome, you get about a 24 diagnostic rate for unsolved cases, meaning if you didn't, they included some knowns, and about half if you look at all comers. And again, at whole exome, just for complex disease in general, they have about a 40% hit rate in general for making a diagnosis. And so really very nice and very non-invasive. And now it's become the standard to include the mitochondrial DNA. In about half of the labs in America that are doing exome sequencing, the mitochondrial DNA is included automatically. Over the last year or so, that's been a nice change. But you have to look where you're getting it done. So in the clinic, we do a very careful clinical evaluation, histories, exams, pedigrees, metabolic screening studies. Tissue analyses are of course still done, but they're no longer the front line test. Sometimes we actually do them after the genomic data to better understand, the implications of a specific gene or variant that we've seen. And of course, we do mitochondrial DNA whole genome analysis, both next-gen to look for sequence variants and deletions, and qPCR to look for copy number. We do a SNP array, and we go very quickly to exome. And the legal system in America is in a battle over which insurance companies will or won't pay for this, but it looks like the patients are winning, and over time, more and more companies are paying for it. So how do you get from all of that complex clinical care, and how do you explain to a given patient, what's known, what's not known, when the line is constantly moving? if Dr. Zeviani publishes a new gene, the next week the clinical lab will offer that as a clinical test. The week before it was research. So the line between clinical care and research is very, very, is a changing target. So in America, I myself have my own data repository at my center and my own tissue repository, probably like a lot of the centers in Italy. And the issues are that it's very time intensive, very detail intensive, and very labor intensive to maintain your own biorepository. You need to get your IRB approval and oversight. You need to have a database to manage it. You need to get the tissues and establish the cell line. It's all very costly to obtain and to maintain, but there's very high potential payoff. You know your subjects very well, much better than some bioinformatician in some diagnostic lab. You know every detail about your patient, and you have a very rich source of material for testing, again, for the genetics and the understanding of the mechanisms and coming up with therapies. This is outdated by a year or two. We have probably twice as many now, but we create lymphoblastoid cell lines on every patient that we see. We create skin fibroblast cell lines on many over time, if not all. We learn their genetic diagnosis, and we bank DNA. And we also take leftover tissues when they're there. So we have all of those within our center, and on people with lots of different genetic diseases. We use many different systems to coordinate our data. So there's a LIMS system to track our data. We use REDCap. I don't know if you guys have used REDCap. This has taken over America. It's a free system called our Research Electronic Data Capture System. The University of Vanderbilt created it. And basically each institution creates its own instance of it. And it's a very simple tool where you can design questions to track what's in your database, both samples-wise and phenotypic information-wise. So very nice. Clinical data, we have a lot of electronic data healthcare systems. Epic is the most common. And we have different systems for clinical trial management and to explore your data. So a lot of very data intensive to be able to keep all this. This is an example of what REDCap is. It's the same login wherever you go. And then you can very quickly create queries that are applicable. So was it a definite mito disease, a probable, possible, unlikely, and it's all de-identified, it's just codes. This is an example of different survey, or not survey, but data capture instruments we created. So a review of systems can take a genetic counselor just minutes to go through and record the data, developmental history, when people rolled or sat or crawled, again in just moments. Our whole sample registry now, my entire laboratory to find a sample in a minute, what box, what sample, what passage, and it was all created using free software. But there are lots of issues if I open up my sample repository and my data to other investigators. At CHOP, I have many, many people from many, many departments and divisions who access these samples and these data, once we know that they're allowed to, but there are trust issues. You know, when you just get data out of any registry, who maintains it, who put it in, how accurate is it, what's the quality of the samples, mito disease cell lines don't grow well, how were they maintained to begin with, what are the incentives why people should contribute, who gets to control the data, who gets to decide you know, who's carrying the burden and who gets the benefits. A lot of more senior investigators feel like they've invested their career in this, why should they give it away? There's also, again, the governance, and then just the transparency, and some of these systems are more or less easy to use than others. And then can anybody get them, and how? So what's a big advance in America is taking like what's at our center, and they've incorporated, and they're now up to 17 different sites, mainly in the US, and then Mark Tarnopolsky's group in Canada. And this is run out of Columbia, but it's really a National Institutes of Health government-run contract to create the North American Mito Disease Consortium. The goals are threefold. Is to create a mito disease patient registry. As of June 1st, I got the update. There's 790 patients in that registry, and there's 3 natural history studies going. It serves as a patient database for clinical trials. Pharmaceutical companies can call NAMDAC and ask questions about the database. And there's also detailed phenotype required that the physician who saw the patient puts in, or the genetic counselor associated with them, and there's payment for every single patient that you put in. So there's a huge incentive to do it, and there's funding to support it. In addition, there's a specimen and DNA biorepository that has been less successful. There's probably about 50 samples in it to date, and part of it is just the time that it takes to send samples there. They would accept any tissue type. So in addition to the North American Mito Disease Consortium, which is mainly physician-entered data and a partnership between the Mito clinical centers in North America, the NIH, and the UMDF. The UMDF, as you heard moments ago, also has a patient-based registry. This launched last September. They're up to about 1,100 registrants. It's only in English right now, but it's not considered the UMDF registry. It's considered the MDCR, or the Mitochondrial Disease Community Registry. And the idea is that patients and caregivers and families provide the data. They don't have to have a confirmed diagnosis. And the goal is that the patient data collected over time will improve diagnoses and the ability to develop treatment. And the registrants, the patients and their family members absolutely control who sees the data. And this is a big issue. If somebody gives their data to me, is it my decision or is it their decision who else can see it? Ultimately, I think if you think about it, it should be the patient's decision. But they actually can choose these things. And there's, if you go on the website, it's very simple to use and it's very easy to send out questionnaires or blasts, like if you want to know what vitamins and supplements people are taking or what symptoms they're having or what have you, and they can change their mind at any time. So the key points that Dr. Phil Yeske, who's the Science and Alliance Officer for UMDF, generously shared with me to inform you about this registry, because I think it's a big thing you guys want to hear about, is that it's sponsored by UMDF, but it's meant to be a community asset. UMDF has pledged to continue to support the monthly fees to maintain this registry, but, and to serve as a guardian, but they actually don't know anybody's name or any information about anybody who's put in. It's kept by a third party, and so only data output is put in. The patients themselves are not recognizable. And the patients can come from anywhere in the world. So this is, I think, a nice platform that could be a way to do international collaboration. It's meant to collect health information over time. It's not meant to replace any other registry. And once again, it's critically important that the patients control the privacy settings of who they want to share it with and when. If somebody wants to share their data with the whole world, they should be enabled to. And if they want to share it only with their doctor, they should be enabled to. And it's allowed them to do it. So I think I mentioned that there's a little over 1,000 accounts. There's been surveys, and there are going to be fact-based. Okay, sorry. I'm not even counting. So anyway, so over time, the goal is to also bring in genomic data, electronic health records, and new international engagement. So I definitely want to encourage, and I know you already are talking, the Mitocon and the UMDF, I think, has much of the same goals. And so how can you get there, I guess, as quickly and effectively as possible? One of the big areas that as a geneticist I felt was important to make available around the world is a common mitochondrial disease sequence data resource. Just the phenotypic information about mito diseases is confusing. Really having accurate understanding of the genomic basis of diseases even more so. And we felt, a group of mitochondrial physicians and researchers, that an international database was needed to unify the genomic data to facilitate diagnosis, but also understanding of mitochondrial disease. Things like having the data right there when you want to look at modifiers, or you want to look if somebody has a variant that might put them at extra risk or make them a better candidate for a clinical trial. So we think genomic data is incredibly useful for many reasons. This is made for researchers and clinicians right now. It is not made for families. We're hoping in the future to be able to do that, but it really takes a huge knowledge of genetics to be able to use this properly. Here's the website. I'd encourage you all to look. It's free. Any academic user can get on and use it. If you don't log in, you only see 5% of the data. If you do log in, you see 100% of the data. And so we want diagnostic labs to use it, clinicians and researchers, and we have many diagnostic labs in the U.S. supporting the development of this. It's a flexible web-based suite of software that's accessible. You can look at it right now. You can get on the internet, and all the data is maintained in real time. It allows you to exploit allele frequencies, which is very important. So how common is a given variant in a specific population? And we are in the process now of being able to add in other data types, so phenotype data, laboratory data, protein, RNA data, etc. And we think this will really accelerate the pace and accuracy of known and novel gene discovery in mito disease. What we're able to do now is to have people put data in an aggregate And what we're working towards is to have patients tell us themselves how they want their individual data put in and held. So right now, individual patients cannot, but we're working on the consent processes so that hopefully soon they can. This is what it looks like if you log on. It's called mctr.org. The big theme is that when you hover over any word, more options appear. You log in from the top. Once you have a login, it allows you to use every resource in here. There's a lot of data that goes into this. We've had people from around the world collaborate and share data. A lot of evolutionary biologists, mitochondrial DNA experts, and nuclear gene experts. And so all this data is in the back end, and then when you log in, you have many different ways that you can utilize this data. One is a browser that's customized for mitochondrial biology and disease researchers. There's many tracks of information that have been established, things like the pseudogenes, the nums, the variants that are seen in any one researcher's lab, whether a gene is a known mitochondrial disease gene, and the entire mitochondrial DNA is visualized, which is not on the regular NIH browser. This is a very important resource, I think, that patients too will find value in. It's called the LOVD, or Locus Specific Database, and we host precise information about every disease, gene, and variant related to mitochondrial disease. We host 1,350 or so different genes. And so when you log in, you can suggest other genes that should be added. You can comment on the authenticity of a specific variant or the call, or whether it's pathogenic or benign, and it tracks you. You can only see these things if you're logged in. And it keeps a list of all the phenotypic information available about every disease. GenMap is a tool. The name is changing to Genesis, but the concept is the same. You can upload your own sequencing data, whether it's a gene panel, one gene, the whole exome, the whole genome, and you can mine it yourself. Without using code, but it's very clickable, easy to use format. It's incredibly fast. It does queries within seconds. We've also created a central portal that provides a link to all available tools for mitochondrial biology and disease analysis, so I'd encourage you to check it out. What we're doing is implementing a few more pieces, such as a global universal ID. So for example, I've had a patient referred to me from Italy with Lee syndrome, whom I follow very closely, but if I entered her data and somebody in Italy entered her data, that data would be overrepresented. So that patient, when their data is entered, has to have one universal ID to keep the data clean, so to speak. We're working with the NIH to integrate the common data elements of what types of phenotype fields should be captured. We're working with what I said was the Genesis project to build in other data types, and we're working with NIH because we really want this to be useful for clinical diagnostic laboratories. We're also working on the ethical use and oversight committees and processes. We have a tutorial that's live online that you can go through from the main page, and we've done surveys now at national meetings. We've done 2, we have one more coming up at UMBF in a few weeks, teaching researchers and doctors how to use this. So far, it's been very positive, what people like and what aspects they think are the most useful. This is a link. This is a Google Analytics link, people using it. So this is as of a month ago. There's 1,000 users from all over the world every day. So I'd encourage you to use it, and better yet, critique it and tell us what needs to be fixed and improved, and that's how it'll get better. And there's been a lot of people from around the world working on this effort, and so we of course love to include the Italian data sets. Of course, everything is properly acknowledged. And so in the last couple moments, so what do you do with the patient specimens once you have them and you have all of this data? One, of course, is you validate new genes. You find secondary cases, and then you need to do understanding, was the data real? You Sanger validated. You do functional studies. Some functional studies in America are clinical, most are research. I think in Europe, still most are research. So what do you have to do to prove that a new variant, either in a known gene or in a new gene, is the cause of your disease? There's a lot of work that needs to be done, including gene rescue when it's loss of function and making cyber lines when it's a mitochondrial DNA variant. Here's just one example of a girl with Lee syndrome I followed for about 7 or 8 years before we figured out what her disease gene was. When you do her exome, you come with 150,000 variants. That's how many we all have. When you ask how many are bioparentically inherited and look like they're changing the protein, about 800. And when you sort it by what's mitochondrial and what's new, you got down to 1 gene. You prove it. This is what next generation sequencing sequencing data looks like. You prove it the old-fashioned way, you standard sequencing. And then you start to do functional studies to look at the mitochondria, to look at the mitochondrial function. And at the end of the day, we were absolutely wrong. I was going to call her in and tell the family this was the diagnosis, and they were considering testing the next baby for this, because we had filtered as just known mitochondrial disease genes. What happened is, in the time I had looked, what were two novel variants. One turned out to be present in 1% of the population. It was much too common to be believable. So we opened up the filters and we found it. I'm working with Dr. Zeviani and a bunch of other people in the Middle East and in Europe. We put together a cohort of about 8 or 9 families, and they wound up having an FBXL4 disease gene. Now there's about 20 or 30 families known, and we're sure that this is the cause, although we still don't know exactly what the gene is doing. So this is a wonderful example, and I think there's many of these now, of how putting data together is valuable. The other thing, of course, is that you can start to understand once you have these cell lines, what's wrong, either within a common disease or within different forms of disease. So this is a publication from a couple years ago where we looked at fibroblasts or skin cells from patients with a dozen different mitochondrial diseases. And we said, what do they have in common? And for example, the ribosome, or what makes proteins in the cell, is the most upregulated pathway, and it's very significantly changed. Other signaling pathways as well. And when we went to look at it further, and we compared it to muscle results, we realized there's a difference in translation. There's making proteins in the cell, and there's making proteins in the mitochondria. And the signals to do this are very much changed in mitochondrial disease, so that the ones in the cell are being turned on, and the ones in the mitochondria are being turned off. And when we looked at databases, we saw that this was the same whether you look at complex 5 deficiency or even MELAS. And so what it led to is our understanding is that there's a lot of differences in how the cell handles this ATP deficiency, this mitochondrial dysfunction, and the NAD/NADH alterations. And depending on the tissue demands, how much energy it needs versus changing things like transcription and translation at a very basic level, very consistently. We've done meta-analyses now. I've almost never seen a mitochondrial disease or model that doesn't change transcription and translation in a very basic way. And we realize that there's signaling pathways that many of you have heard about that are involved in affecting these changes. And a lot of us focus on one or another at any one time. But what we realize is that there's actually a network, and that they're often all changing. You can't change one part of it without changing the other. And so this is cell lines from 3 different patients of 3 different genetically confirmed mitochondrial diseases. And these are the complexes that are deficient. And you can see that there's different patterns of how these signaling molecules are turned on or turned off. And what does this mean to us? Well, it means to us like what you heard about this morning with Dr. Zeviani and rapamycin, is that rapamycin, one of the things that it's doing, or the thing that it is doing, is shutting off mTOR. And the mTOR pathway directly regulates 2 really important things. One is translation, the ability of cells to make proteins, and the other is cell death through the process of autophagy. And so we just had a paper that came out today, as a matter of fact, online. But basically what we realized is that rapamycin's okay, but if you go to the processes that the mTOR pathway is trying to regulate, if you directly, more effectively shut off translation a little bit, not completely, or the cell death pathway, autophagy, is much more effective, incredibly effective at maintaining cell health and viability in mitochondrial disease. And this is rotenone, so this is the number of cells that are dead. If you use a molecule called cyclohexamide, which shuts down translation about 60%, you get 100% rescue of cell viability. And it doesn't matter what cell type you're looking at. And you actually get a restoration of mitochondrial function as well, regardless of which part of the respiratory chain you inhibit. And this just goes back to the FBXL4 cell line from that little girl that I told you, that we had the right gene when we all worked together. Her cell line superoxide levels come down when we treat them with cyclohexamide, and her amount of mitochondria go back up. So having these resources is very helpful to figure out what the right therapies are, and this is, I think, my last slide. That basically when you think about the mitochondria, you have to think about what pathways are changed and what processes those pathways are trying to change, what they're trying to achieve. And that once we understand that, we can use drugs that more directly and maybe more effectively improve the health of the cells where the mitochondria are defective. And We heard a wonderful talk this morning about niacin as well, and we had published this in a cell line of a patient with complex 1 disease dural Leber's mutation who actually has LAN plus or Lee syndrome. And not only was her sirtuin levels altered, but her mTOR activity and her AMP kinase activity was altered, and all that got better when we treated her with niacin. And she had a total cellular, forgive me, deficiency of these redox molecules. And nicotinic acid made those better, and it also made her cell respiration better. And so to conclude, we believe that characterizing and treating the central signaling alterations that are in a cell or the physiologic processes that they're maintaining offers a personalized therapy depending on what's the prevailing pathology in a given patient to treat them. And that therapy, you'll see, there's nothing on here that's in the traditional Mito cocktail, right? There's a whole new approach to therapy. And so we're hopeful that by understanding what's going on in each patient, we really can come up with some effective therapies for each person's disease. And I'm hopeful that by doing this, it's going to change the way we do clinical trials. So we'll hear today that clinical trials are coming, and nothing can be better. But the truth is which ones do you pick? There's only a limited number of patients. How do we decide which clinical trials to push forward? And so I would argue that once you know the disease and the outcome you're going for, and you understand what the treatment options are, that you stop. You don't go right to clinical trials. You stop and go to the laboratory, and you take these patient cells or these models, and you understand which ones are most effective for those patients, and then it's those patients that you put in the clinical trials. And so you increase the likelihood that you'll get positive clinical trial results, and you'll be treating the right patients with the right drugs. That's it. Thanks, everybody.
Excellent talk. You are showing us it's very important to collaborate, starting for maybe a very far point, but you got the results that are clearly suggesting for future improvement of this data. The presentation is open to the discussion.
Yeah. I have the impression that your approach is very fruitful. I don't know why you said that CPT is not the mitochondrial disease, it's the Indian mitochondrial disease.
My definition was respiratory chain disease.
Excuse me?
In that paper, we were thinking about respiratory chain disease. So most people.
You should then say, I include only Oxford.
Right. So I think this is also a really big question. So we've never had a meeting where we discuss what is a mitochondrial disease where anybody agrees. I think what people mean when they refer patients to us is this an Oxford disease. I think that's what's in people's heads. We just published a paper that just came out that showed desmin deficiency. It anchors mitochondria, right? Dr. Gazy put together a wonderful series of reports, and this person had very little mitochondria. Right? And that makes sense because desmin is an anchor for the mitochondria. Yeah. Nobody thinks of desmin as a mitochondria. Yes.
What is the criteria?
Well, so I think the most important thing for me is to come up with the genetic etiology in a patient and then categorize them later. So that's what I'm trying to say is I think historically when you started with the biochemistry, people were looking for the genetic causes of Oxford disease.
Yeah, but I have the same problem you can have. So it's not the problem.
So, any more questions? Okay, thank you.